Hello, my name is Paul Riley. I am the Portfolio Development and Engagement Scientist from Stago North America, from the Stago Scientific Marketing Team, and I am joined today by my colleague, LaShanta Bryce. In our last edition of our Stago Scientific short series on lupus anticoagulant testing, I interviewed Claudia Escobar from our technical training team, who discussed in detail the different methodologies involved in lupus anticoagulant testing. Now we will dive into the clinical guidelines available for coagulation labs interested in implementing this panel of tests, or for those currently testing but who may need to update their practices.
Hi, everyone. My name is LaShanta Bryce, and I'm the Scientific Engagement and Clinical Education Scientist for Diagnostic at Stago, also a part of the Stago Scientific Marketing Team. There are several guidelines available for lupus anticoagulant testing. However, this presentation or podcast will focus on the International Society on Thrombosis and Hemostasis and the Clinical and Laboratory Standards Institute guidelines. The 2009 ISTH and 2014 CLSI guidelines have been very useful in achieving greater uniformity in the performance and interpretation of lupus anticoagulant testing. ISTH has since then authored new guideline updates as a part of the Scientific and Standardization Committee's commitment to continuous improvement, while CLSI plans to update their H68 laboratory testing for lupus anticoagulant in the near future.
Yeah, that sounds really interesting. So what do the 2009 ISTH guidelines mainly recommend for clinical labs performing lupus anticoagulant screening and diagnosis?
In short, the 2009 guidelines from the International Society on Thrombosis and Hemostasis on lupus anticoagulant testing in the clinical laboratory recommend use of two different phospholipid-dependent clotting tests, the APTT and DRVVT assays. The recommendations were based on expert recommendations and literature review by international key thought leaders in the clinical coagulation laboratory. We discussed the two tests in previous releases of the Stago Scientific short series, pointing out both assays target different parts of the overall coagulation pathway, giving information to clinicians regarding which parts of the pathway are inhibited. The recommended test must be run as a paired screen confirmation combination using low and high phospholipid levels, with the low levels providing sensitivity to detect the lupus anticoagulant and high phospholipids neutralizing the lupus anticoagulant effect.
Yes, according to the 2009 ISTH guideline, the lupus anticoagulants, or LA, are confirmed if the result exceeds the positive limit for an APTT-based LA confirmatory assay, the hexagonal phase phospholipid neutralization assay, or if the result for the DRVVT normalized ratio result exceeds the cutoff determined by the lab. LA is diagnosed if either the APTT or DRVVT-based LA confirmatory tests are positive, and if the result is still positive in a separate set of tests run 12 weeks following the initial testing panel. And then additional tests should also be ordered according to the ISTH guidelines, which include the anti-cardiolipin or anti-beta-2 glycoprotein 1 tests, which are usually sent to a reference lab to help determine if the patient has additional positive test results, which would then contribute to the overall patient risk profile. The 2009 ISTH guideline, as you might remember, helped to greatly simplify the recommended tests and reporting methods based on available evidence at the time from analyses of proficiency testing results over multiple years.
The recommended order of test from the guideline is screening first using a sensitive reagent containing micronized silica, then mixing studies, followed by confirmatory tests with high phospholipid levels. In addition, if ratios are reported, such as those seen with DRVV testing, the denominator should come from normal pool plasma. When it came to testing patients on anticoagulants, mainly unfractionated heparin and vitamin K antagonists, such as warfarin or Coumadin, the guidelines recommended interpreting results with caution for patients on unfractionated heparin. For vitamin K antagonists, the recommendation was to perform testing on undiluted plasma if the INR was less than 1.5. If the INR was greater than 1.5 but less than 3.0, the recommendation was to mix the sample with normal pool plasma first before testing.
Yeah, it sounds like there's some differences between the 2009 ISTH guideline and the 2014 CLSI guideline. What are those main differences in your opinion?
One big difference was the testing order. The 2009 ISTH guidelines recommended screen, mix, then confirm. The 2014 CLSI guidelines recommended screen, confirm, and then mix. In addition, the 2014 CLSI guidelines also recommended if ratios were reported, such as those seen with DRUV testing, the denominator should come from an established reference interval, whereas the 2009 ISTH guidelines recommended using normal pool plasma.
Hmm. And then what if the APTT results is elevated unexpectedly at the time of screening?
Often, oftentimes the first step is to repeat the test, which is usually followed by clinical judgment, which may include consultation to the laboratory service or to another clinical service for further guidance. Ultimately, the clinician will make the decision on which follow-up test to perform based on patient symptoms, history, risk of thrombosis, and occurrence. Common follow-up tests may include an APTT mixing study, factor assay analysis, and/or lupus anticoagulant testing.
Okay, thank you so much, LaShanta. This concludes our initial discussion of the LA testing panel and the clinical guidelines. Let's continue this conversation in more depth in our next edition of the Stago Scientific Short Series.
Thank you.